Hello friends, good afternoon. Welcome to some biology class. Friends, today we shall discuss about the structure and function of phytochromes. Phytochromes are a widespread family of red and far red responsive photoreceptors. They were first discovered in plants where they constitute one of the three major classes of photomorphogenesis regulators. All phytochromes utilize a covalently attached Billion chromophore that enable photoconversion between red light absorbing form and far red light absorbing form. Phytochromes are thus photoswitchable photosensors. Canonical phytochromes have a conserved N-terminal photosensory core and a C-terminal regulatory region that typically include a histidine kinase related domain. Phytochromes are present in plants, bacteria, and fungi. Phytochromes were first discovered in 1959 by Sterling Hendricks and Harry Borthwick. Phytochromes are chromoprotein. Their molecular weight is 250 kilodalton. It is a homodimer consisting of a protein part and a non-protein part. The protein part is composed of two identical polypeptide chains known as subunit A and subunit B. Each subunit is covalently associated with a billion chromophore that absorb red light and far red light. Each subunit has two domains. First domain has photoreceptor activity and the second domain has kinase activity. This figure shows the domain structure and chromophores of 5 different classes of phytochromes. PHY is the phytochrome present in plants. CPH1 is present in cyanobacteria and BPHP is the bacterial phytochrome. Each subunit of these three different classes of phytochromes share a similar architecture consisting of an N-terminal photosensory region and a C-terminal regulatory region. The N-terminal photosensory region consists of three conserved domains. They are P2 domain, P3 domain and P4 domain. P3 domain is covalently associated with a billion chromophore. The C-terminal regulatory region has histidine kinase or histidine kinase related domain. Plant phytochromes have an additional N-terminal extension known as P1 which inhibit the dark reversion. In addition to this, the plant phytochromes also have two additional regulatory pass domain which is important for nuclear localization. Another class of phytochrome present in fungus. The fungal phytochrome has a distinct N-terminal extension and have an additional C-terminal response regulatory domain. And another subclass of phytochrome is present in some cyanobacteria known as CPSP2. So friends, this is all about the domain structure of 5 different classes of phytochromes. Now we shall discuss about the structure of plant phytochrome. Plant phytochrome is a homodimer. Its molecular weight is 250 kilodalton. It consists of two identical polypeptide chains. Each polypeptide chain is covalently associated by a thioester linkage with a abyline chromophore. In plant phytochrome, the chromophore is phytochromobilin, which is synthesized in the plastid and is then leaked into the cytoplasm, where it is associated by the thioester linkage with the polypeptide chain. Each polypeptide chain has a C-terminal regulatory region and an N-terminal photosensory region. The N-terminal photosensory region has three conserved domains. They are PAS domain, GAF domain and PHY domain. PAS domain stabilize the PFR form of phytochrome. The GAF domain contain a conserved cysteine residue with which the abyline chromophore is associated by a thioester linkage, and the PHY domain participate in the synthesis of phytochrome receptor proteins. The C-terminal regulatory region has also several domains. HKRD is the histidine kinase related domain which acts as a serine threonine kinase. The plant phytochrome has two additional regulatory pass domain which is important for nuclear localization, and the pass domain also is responsible for phytochrome dimerization. So the plant phytochrome consists of two identical polypeptide chains, and each polypeptide chain is associated with a abyline chromophore. As already mentioned, phytochrome is a red and far red responsive photoreceptor. By undergoing interconversion between two different forms, it brings about morphogenetic responses in plants. Phytochrome exists in two interconvertible forms. They are PR and PFR form. PFR form is biologically active, whereas PR is biologically inactive. In PR form, the abyline chromophore is in cis configuration, whereas in PFR form, the chromophore is in trans configuration. So during the interconversion between PR form and PFR form, the abyline chromophore undergo cis-trans isomerization. The PR form absorbs red light strongly at 660 nanometer and undergo quick conversion to form PFR, which is biologically active and induces morphogenetic responses in plants. Similarly, PFR form is also converted into PR. PFR absorbs far red light strongly at 730 nanometer to form PR. This process of conversion of PFR to PR in the presence of far red light is a quick conversion. In addition to this, in darkness, PFR is converted slowly to PR. So this interconversion of phytochrome between PR and PFR in response to red light and far red light is called as photoconversion. So by undergoing photoconversion between two interconvertible forms, phytochrome brings about morphogenetic responses in plants. In addition to induction of flowering, phytochrome is involved in various morphogenetic responses. These responses can be divided into two different groups. They are slow responses and fast responses. Slow responses include seed germination, stem elongation, hook opening of plumule, cotyledon and leaf expansion, flower initiation and de-etiolation of seedlings. And the fast responses include leaf movement in Minusa pudica and chloroplast movements, surface potential changes and also membrane potential changes and ion flux. So friends, this is about the structure and function of phytochrome. For more updates, please subscribe my YouTube channel Sam Biology Class, and also please share your comments. Thank you very much.